All right, thank you all for coming today. I'm going to talk about some of the research I've been doing on plant-associated fungi and their interactions with bacterial communities in drought-affected soil. I want to thank a lot of people who have helped me with this research and also my funding, which is from the Department of Energy. And before I get started, I wanted to just spend a minute talking about the motivation for my work, particularly because we're agriculture. I spent several years working on farms prior to going to graduate school, and we were spending thousands of dollars per acre on microbial inoculants with the hope that we would improve crop nutrition and stress tolerance using those microbial products. But we really didn't have much information about how those products were working and how to test whether or not they were working. And so that was a large part of my motivation for seeking education in this field. And I was hopeful that maybe we'd be able to harness plant microbial synergies for more productive, resilient agricultural systems. And in particular, I became really interested in plant-associated fungi. So these are arbuscular mycorrhizal fungi colonizing a root. On this image here, you can see the branched arbuscule structures, which is where resource exchange occurs between the plant and the fungus. And there's been quite a bit of research showing that this can really benefit crop nutrition and stress tolerance. And more recently, there's also been more and more work showing that this interaction doesn't just depend on the plant and the fungus, but also on how the fungus interacts with the rest of the soil microbial community. So these are thousands of bacterial cells growing along a fungal hypha. I'm really interested in trying to understand which organisms are there, what they're doing, and what the ultimate effect is on plant and ecosystem function. And you know, so in my postdoctoral work, one of my focal points was trying to understand how these interactions influence microbial response to environmental stressors like drought. That drought is predicted to increase in severity and frequency. We know that it influences microbial community structure and function. And we also know that AMF or arbuscular fungi can support plant function during drought. And we know that the fungi themselves are releasing different exudates and carbon out into the rest of the soil. And we suspect that that influences the rest of the soil microbial community, but we don't really understand exactly how it affects the microbial community, its composition and its activity. And so that was one of the main goals of the experiment that I'll describe today. So our question was how these plant-associated fungi mediate microbial responses to drought. And based on this understanding that drought influences microbial structure and function, and that plant-associated fungi release exudates into the soil and probably alter the soil, we hypothesized that these plant-associated fungi would mitigate the effect of drought on soil microbial communities, and also that microbial responses to different fungi would be distinct. And so in order to investigate this, I used two different plant-associated fungi, Rhizophagus irregularis on the left, which is an arbuscular mycorrhizal fungus. It's found all over the world. It's an obligate symbiont, so it depends on its plant host for carbon, and it has a reduced enzymatic repertoire compared to a lot of other fungi, such as Serendipita besseyi, which is the other fungus that I used. Sebaceinalis fungus. It's a facultative symbiont, so it can get carbon from the plant or from detritus that's in the soil, and it has a wider enzymatic repertoire. And in order to investigate those fungal interactions with the plant and with the soil microbial community, I grew Panicum hallei plants in these growth chambers shown here, with and without those two fungi, in a soil that we collected from Oklahoma, which is part of Panicum hallei's natural range. And then I grew those plants with the fungi and with the soil microbial communities for up to 12 weeks. And I used 25 micron mesh to exclude roots from a component of the soil, but allow the fungal hyphae in. And so that way I was able to investigate processes that were going on in that fungal hyphosphere, or the region around the fungal hyphae, and distinguish that from the rest of the soil. And then I grew these plants under either water limited or water replete conditions in order to investigate how water limitation would influence those interactions. So there are a lot of pieces of this experiment that I won't be able to describe today, but if you're interested, please feel free to reach out later. Okay, so with this question of how plant-associated fungi mediate microbial to drought, one way that we can assess that is using 16S rRNA gene profiling, which gives us an indication of the bacterial community composition in the soil. And so this is an ordination plot here shown on the left that includes all of the 16S rRNA gene sequences that we were able to sequence in the soils that were maintained under different fungal conditions. So those are indicated in the different colors, and under the different moisture conditions, which are indicated with the different shapes. And you can see that at 12 weeks, which is what's represented here, there is some differentiation between the microbial communities present under these different conditions, but it's actually, there's a lot of overlap, and it's kind of hard to distinguish those differences. And one reason that this could be is that this kind of traditional 16S rRNA gene profiling, we're sequencing everything that's present in the soil. And so we often get a pretty big background signal in soil from microbial organisms that might be dead or dormant. And one way that we can try to distinguish the actively growing community from that dead or dormant community of DNA is by using a method called stable isotope probing. And essentially what we're doing here, this is a simplified schematic, so there's it's a little more complicated than what I'm representing on this slide here, but essentially what we do with DNA stable isotope probing is use an isotopic tracer, and as microorganisms grow, they incorporate that heavy isotopic tracer into their DNA, and then we can use centrifugation and differences in density of the DNA of organisms that take up that isotope to distinguish organisms that were actively growing from organisms that were not growing. And so at the end of the greenhouse experiment that I described, I incubated soils with either 18O water or 16O water, and I used this approach to identify bacterial taxa that were actively growing under these different conditions. And so if you think back to this ordination plot that I showed earlier, this is the plot on the left based on the bulk community. And this plot on the right is a plot that I made by filtering our data set based on the results of that stable isotope probing. So this shows just the actively growing organisms, and you can see that we're able to distinguish the effects of those treatments much more we're able to distinguish the effects of those treatments much better when we use that stable isotope probing analysis to filter out organisms from those that were active. And so in the next several slides, I'll just show results based on that stable isotope probing analysis. So here we have an index of diversity on the y-axis, and you can see in the uninoculated soils, there's a significant decline in bacterial diversity following water limitation. Whereas when we inoculated the soils with the two different mycorrhizal types, we see a negative effect of water limitation on bacterial diversity. And similarly, here on the y-axis, we have 18O atom percent excess. So this is a metric of bacterial growth potential. We see a negative effect of water limitation on bacterial growth potential in uninoculated soils, but in the presence of the two fungi, we don't see a negative effect of water limitation on bacterial growth potential following water limitation. So these are what I showed just now were bulk metrics of microbial response. But one of the great things about DNA-based methods is that we can use them to look at specific responses of amplicon sequence variants. So in the next few plots, I'll show the response of specific ASVs. So each ASV will be represented by a different bar. And here I calculated a growth response ratio to indicate whether those taxa were more active under under soils inoculated with the plant-associated fungi or not. And so if that bar falls above this horizontal line, that organism was more active in the presence of the fungus, and if it falls below that line, it was more active in the absence of the fungus. And so we can see that kind of across the board here, most bacterial ASVs were more active in the presence of the fungi than in their absence. We also see that there's a different effect of the fungi. So we see that some organisms respond positively to one fungus, but not to the other And we also see that overall bacterial response is more positive in the presence of the Rhizosphagus irregularis than in the presence of the Serendipita besseyi. And one thing that I think is particularly interesting is that a lot of these taxa that responded positively to the plant-associated fungi are taxa that are generally considered drought susceptible. So it seems like somehow the fungi altered the way that allowed these organisms to maintain activity even under water limitation. And so kind of bringing this back to the bigger picture of agriculture, I think that there's still a lot of questions that we have that we need to work on, but we do have some evidence that these plant-associated fungi supported bacterial growth and diversity following water limitation, and that they might promote biotic resilience and recovery following stress events like drought. And whoop, that just exited me out, so I guess I'm out of time. Thank you all for your attention.